Hi, everybody. So I am Aritra Bose. I am a research scientist at IBM Research, and this is some of our preliminary results that we have been getting applying quantum random walks in biomolecular networks, in specifically in two kind of different algorithms and also applications. And obviously, this is a work in progress. So, like the paper currently is out on this preliminary results, but we are currently doing a lot of extensions to this, just as a disclaimer to start with. So yeah, what we are trying to kind of like do from the onset, and what I think many of you who are interested in this NetBio community do, is like modeling multicellular networks or modeling, in general biomolecular networks. And in that, we try to model like how these living organisms work, and sometimes the cellular microenvironment work, right, in the CCI cell-cell interaction situations. So I will not go into deep detail here, but you know that like there's a sending cell, there's and a receiving cell, so there's a ligand and the receptor kind of binding in that phase, and then there are multiple kind of resources out there to do CCI network inference, like some of them I have kind of like pointed out on the right hand side, that CellPhoneDB, CellChat, there are many of those, and tensor cell to cell. What we are interested in is to kind of trying a more fundamental approach to cell-cell interactions, try to see whether random walks on these cell-cell interaction networks would lead to anything meaningful, and specifically quantum random walks, because there are some inherent constraints in classical random walks, which I will also briefly discuss today. So can quantum random walks be a kind of alternative to infer a different kind of space that the classical random walk is not inferring, and maybe it's seeing something like different when Euclidean distances or any distance measure of your choice. And just we wanted to kind of like start out from that question, right? That was the motivation. And it's just to note that quantum random walk is not the only way to kind of go about developing quantum algorithms for biological interactions. GNNs, and then there are equivalents of GNNs in quantum GNNs, and there are like other kind of CNN and quantum CNN methods which might be also leveraged, and other machine learning methods in general. But we are interested in quantum random walks because of its kind of appeal for interpretability. So just a little bit of unsolicited math, but like bear with me. That essentially what we are trying to do here is distinguish between a very close analog of classical random walks. And there are different kinds of random walks. There's a continuous time random walk and a discrete time random walk. And it's kind of like boils down to the essential difference between continuous and discrete, that, one kind of follows like a clock. If you wait for a certain amount of time and then you make a jump to the next node, and the other kind of follows with like some random distributions. So you have some kind of, you wait for some time and then you randomly jumps to the next node, right? So this is what the walker, we'll call it a walker, the walker decides to do in these different situations. And for whatever purposes, like in continuous time random walk, you can form these like underlying matrix of the graph as a Laplacian. And you just heard Laplacians in the last talk, so I'm not going to go into much of details like what are Laplacians, etc. But the underlying idea is that in quantum, you have these, like on quantum or classical, in continuous time random walks, you have the transition matrices, and those transition matrices come from the graph Laplacian. And in quantum case, it also comes from the graph Laplacian. It's just that you are trying to model it based on Schrodinger's equation instead of some other kind of way of just not projecting it onto any other space. So the only difference is that you replace that kind of M matrix, which is a graph Laplacian, with a Hamiltonian now, which is also a graph Laplacian. But the Hamiltonian follows from the Schrodinger's equation. If you have some idea about quantum mechanics, you can also look it up if you want to. I will not go into the Schrodinger's equation detail of it because it will get to a course of which will take probably a whole day. But you can see the similarities, and they're close analogs. So, and similarly, in discrete time random walk, the walker kind of decides with equal probability whether standing on a node by its degree, which degree or which node it will move to. But in quantum random walk, that is a bit different. So it uses something called the Hadamard operator, which is kind of like an entanglement kind of matrix. I am not showing it here. It is also like, it is like very basic level of linear algebra for Hadamard matrices. So you use that Hadamard matrix as like decision-making system to move to the next kind of, node. Essentially, which the classical random walk would just take a coin flip and then move. And instead of the coin flip, we now do this principle using a similar, replacing a coin flip with a Hadamard matrix. That's a very simple thing to do. And then you have like a tensor product between a shift operator and a coin operator. So essentially the coin operator comes from the Hadamard matrix and the shift operator just shifts it to the next node. That's the only difference. So it might be a little bit more detailed here, so I'll not get into the details and I'll go to the results section, which might be more appealing for the crowd here. So we first looked at the metabolic CCI network. So these are like metabolites. And then we looked specifically at a bipartite kind of structure, because this is kind of a hard problem to solve in general for flows and networks and random walks. So we wanted to see whether the classical random walk would see something different, as I was saying before, when compared to a quantum random walk, right? And it would help us if there are a structure in the data, so in the graph. So that's why we started with like a bipartite or a multipartite structure. So you have the sender kind of cells, you have the receiver cells, and it goes through a bunch of metabolites and sensors, right? So SLC operators. And then the way it is, we were just following this paper's kind of data to build this for brown adipose tissue in mouse. And you can see how they computed. So those are the CCI scores that are encoded in the edges of this network. So then when we apply our discrete time random walk and compare it with the discrete time quantum random walk, we do see in Euclidean distances, after like 5 iterations of both walks, they are seeing a completely different kind of space, right? So I think this is, if anything you have to take away from this talk, you take away that with quantum computing, we essentially unlock a different kind of space of working all together, which is not bounded by any kind of like what the Euclidean distances would see, right? And here you can see on your left-hand side, maybe I can get a pointer here. Oh, I cannot see my cursor, but anyway. So on your left-hand side, you see these 4 blocks, right? And then these 4 blocks kind of loosely correlate to the multipartite structure that we had. But in the right-hand side with the discrete, and you would expect that because it's a multipartite structure. But on the right-hand side, the stochasticity is just astonishing, right? Because it moves, the walker moves around the multipartite structure in a much more free manner than what it did when it did in the classical random walk kind of situation, right? So there is an inherent difference between how the space is inferred and based on which you will kind of make further inference. So similarly, when we did some, like as somebody was saying, like we cherry picked some instances because we didn't do like a thorough analysis or ablation study here. So we cherry-picked some of the things that we wanted to kind of see, whether the CD8 T cells play any role in malignancy in these brown adipose tissues. And we saw that with the quantum random walk, they kind of like, which are key progressors, they go via L-glutamine, which is kind of validated in literature in quantum random walks, whereas when we did the same question or query for the classical, we did not see such an event. And similarly, for gamma aminobutyric acid, GABA, so it's a very common kind of like, pathway that people are interested in to study with malignancy and myofibroblasts in these adipose tissues. And we saw that the DTQRW, which is the quantum random walk, observes GABA as with like one of the malignant cells, which the classical random walk does not do. So obviously, I acknowledge that there needs to be more analysis done to kind of say something more foundational about this. This is kind of, as I said, a bit of cherry picking, but just to give you a flavor of what we are doing, right? And also, just shifting gears to another sort of biomolecular network where we looked at like PPI networks, but we wanted to rank the genes in the PPI network based on like our previous knowledge about GWAS summary statistics, and so that we could do some biomarker discovery. So we could start with a seed gene, and then we can define a target gene, and we can see how many nodes or vertices is that seed gene, like the walker travels, and if they are key kind of genes that are also found in for the same disease. So we looked at neurodegenerative diseases of autism, schizophrenia, and we also looked at asthma. And we defined the seed genes and target genes, like I have said there, with some GWAS catalog, burden test kind of Pegasus analysis, where we looked at the SNP region and then obtained the representative. And also we defined the targets based on just GWAS widely acknowledged threshold of 5 to the power 10, 5 times 10 to the power minus 8. And we looked at these PPI networks for 5 different PPI networks with very, like, they're kind of large, but they also have a lot of like connected components. And we wanted to look at the largest component for our random walk, where it was, there were a lot of fragments in them. But HumanNet and PCNet are one of the ones that are fully connected. So, which is kind of great, and you will show why. So this is a bit busy slide, but like, bear with me, I'll walk through it. So we applied and if you haven't noticed, we have started doing, we have moved away from discrete time to continuous time random walks here, right? And on your right-hand side, we'll start from the right-hand side, right? So there's the comparison between the CTQRW, which is the quantum random walk, with the the classical random walk. And you see that the stochasticity of the operator of the CTQRW is much high. It kind of like, you see a very random kind of curve, but there's a generic kind of decreasing pattern. And in general, we evaluated the rank of the genes of the walker that the walker saw while walking from a seed gene to a target gene, using this measure, which is the average precision at K. It's kind of common to use that. So mean average precision at K, map at K, to rank the genes. So here you would see that the average precision for the quantum random walk was generally high. And this is a bit more robust than the other result that I was showing, because we did some more kind of involved analysis, like perturbing and changing the nodes and see whether the same result holds, and it did hold. Whereas when you compare that particular trend in the quantum random walk with the classical random walk, you do not really see that much of like changes or fluctuations, but it's kind of more stable. But in average, the result is not that great when you compare with the classical quantum random walk. And what is also captured for these 3 different diseases, so the asthma, autism, and schizophrenia, when you look at these plots. So these plots kind of compare the quantum random walk and the classical random walk's performance over these max average precision at K measure. And whatever that is above the diagonal means that it's performing better than the other. So it's pretty easy to follow. And you see that in almost all the cases, the quantum random walk is performing better in ranking the genes, in finding the genes that are between, like, from a seed gene to a target gene. So potentially unraveling key biomarkers that you might find linked to a disease by doing a classical or quantum random walk versus a classical random walk. And that is also true when you move shift the K from 20, when you rank the top 50 genes, also for 100 genes, and we have that in the paper, we don't have it here. But that's kind of like the general idea. So in these 2 examples in biomolecular networks, this is very early days in quantum computing. I'm sure many of you might wonder why is he speaking about quantum computing? But the reason being that we are seeing something different when we explored this space of network biology with quantum methods, and we wanted to report it to you so that you get interested and as a community start investigating, and we get more awesome results and develop new algorithms. So with that, I'll end my talk, and I'll thank my collaborators and co-authors in this talk. So this was a joint work with Algorithmic, which is a company in Finland, and they are also developing very awesome algorithms in quantum. And IBM research kind of funded this project. And with that, we have the paper on arXiv if you want to take a look. And we are currently working on developing the circuits based on these kind of quantum random walk, testing them in real quantum hardware, which IBM has, and kind of benchmarking these results more thoroughly so that we could have a more definitive say in this work. So with that, thank you.
Thank you very much. Can you explain again the seed and target genes for the evaluation? I'm not sure I understood that.
Sure. So the seed genes are where we have the GWAS catalog. So we obtained the GWAS catalog gene, the SNPs for these particular diseases. And then we looked at the region of those SNPs to do a Pegasus kind of burden test, almost analogous to burden test kind of analysis, to obtain a representative gene for that particular region of SNP. So in that region, if the SNPs are very highly kind of enriched, or I should say SNPs are highly associated with the trait, then we obtain that gene as a marker gene, as a representative gene for all those SNPs. So we did not perform at a SNP level, but we came to the gene level, so that because we were mapping that back to the PPI kind of genetic network, so we did that. And then the target genes we selected of all the other genes by doing the same kind of technique. We randomly selected a seed gene and we set another target gene, and we saw that, and we wanted to find how many nodes it would travel, the walker, from this seed to that target, and what would it see differently than a classical walker would, based on like this difference. The idea was that by doing that, you might find out key differences in biomarkers or the genes that both the walkers would see, and we could compare and interpret what and do some biological enrichment studies to understand whether it sees something more meaningful versus a classical method.
Okay, another question here.
Hi, thanks for the talk. I would like to ask at some point demonstrate whether there's some difference when you let the random walks around between the quantum walk and the additional one. Have you compared the stationary distribution between the two types of random walks? Like if you let the system run, where it converges it always?
Yeah, so we let it converge for like 5 iterations. Okay. Yeah.
But then you say you're achieving the convergence rate in 5 iterations.
Yes, for some experiments, yes, that's okay. Right. Yes. Mm-hmm.
But if you let like the system run for 100 or
you can, yeah, and it doesn't, it just gets darker and darker, and like this is, so what we wanted to see is like the first point of divergence starts occurring in just 5 iterations, and if you let it run, you can just it solidifies your understanding about it. Yeah.
Do you have any biological explanation what you are thinking with quantum walks? Maybe you're capturing some biological mechanisms that are happening not certainty, but are happening probabilistic. Have you done any analysis on that?
That's the work in progress right now. Yeah,
okay.
Doing the biological validation. Yeah.
And another question here.
Hi, thanks for the talk. Could you please comment more on the quantum resources, like how many qubits you were using, or how many gates?
Right. So for this one, we did not use a quantum circuit per se. What we used was the fundamental equations and then encoded them, just like a Hamiltonian we would do in a physical system, and just did that. So currently, the current ongoing work, so that's why we wanted to kind of timestamp this and moved on to actually building circuits. So we are doing that right now, and we are looking at potentially around 20 qubits at this point to form some initial experiments in the simulator, which is something that a package called Qiskit has, which is like a software development toolkit for quantum systems. And then we aim to test it in the hardware with much larger qubit space, because now we can run up to like 130 to 150 qubits. So we want to at least test in like 60 or 80 qubits regime. Thank you for that question.
Now, hopefully short question way up here.
Hi, in regards to the circuit that you just mentioned, do you reckon you could actually, if the circuit that you designed can be applicable to other single cell data sets, something like that?
For sure, yeah. It's very generic, yeah. It is not specific to any method or any data per se, yeah.
With that, I would like to thank you, and I'm going to hand out again.
Thanks, yeah.